So this is the vector, right? So I don't know whether you can read these letters. So origin. So maybe for this question or maybe for another question, they might ask you about the vector. We have seen questions they ask about the vector, right? So what questions they can ask from you? Right? Explain the suitability of the vector is suitable DNA molecule to clone your insert. What features it has? If you're going to clone it in a bacterial cell, that kind of question, right? So then, so don't forget this step. Ori is the origin of replication. So replication start from here, and replication initiate from here, right? So imagine this all the vectors has a certain number of copies in each bacteria, right? That copy number, sometimes it could be 5 copies, sometimes 25 copies. Is same as the human cell consists of mitochondria, right? So mitochondria also have circular DNA, as we mentioned. So mitochondria divide, genomic DNA divide only once, but mitochondria, every single mitochondria divide by once again. Then if that cell has 50 mitochondria, then every mitochondria divide, right? Then you have 100 mitochondria, right? So then the cell divide into two daughter cells. Then 100 out of 100 mitochondria, 50 go to one cell, 50 go to other cell. So that is what you call copy number, right? Because mitochondria also have plasmid inside, plasmid like. It's not exactly plasmid; it's a plasmid like circular DNA. So here plasmid, the same thing happen in here, right? So copy number decide by origin, because higher the copy number sometimes good for us, we get a good yield, right? But it depending on your gene. So if your gene is sometimes when the gene expressing low copy number is more stable, right? If insulin gene is no problem, but some gene like a toxic gene, right? For example, I cloned one toxic gene from Salmonella in E. coli. Then I found no result at all. None of the bacteria is having bearing. So I just discussed my supervisor. So what happened? This host cell is the appropriate host cell for this plasmid. Why once I transform, no single cell is there, no recombinant cell there. And then he said, "Hey, what is the copy number?" I say 25. He said, "Maybe it's too much. Go for low copy number vector." So then I went to copy number 5. Then I found the genes, because if that gene expressing, if it is toxic gene, the host cell cannot tolerate that. Yeah? So likewise, you can have the ORIT decide the copy number, and selective marker, which is what, which is ampicillin or any antibiotic marker can be there, uh, and promoter gene. What's the purpose of having good promoter gene? Manufacturer put a good promoter gene there because the promoter gene encourage this plasmid, encourage the cell to amplify or to express genes after the promoter, right? So express the gene after the promoter. So yeah, this is DNA. So the first step of the gene expression is transcription. So transcription is initiated by a strong promoter, right? And when the transcription initiates, so RNA polymerase enzyme join here, and they amplifying, so amplifying, extending the RNA, copying all this DNA, copying all this DNA, and which include your insert as well, which include your insulin gene as well. And finally, they attach poly A tail into that. Right? So can you tell me why it is poly A tail there? Yeah? Because this is transcription, right? So poly A tail become poly T tail when it become mRNA, right? What is complementary to A? T. So imagine it making a mRNA from here to here. The last bit become T, 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 T. Yeah. So RNA polymerase make T tail at the end. So T tail is important. Why? Because when it's going to translation later translation happen. When it is going to translation, ribosome can understand that is the end of the gene, right? End of the gene, always mRNA has poly T tail. Yeah. So we know that poly T tail, right? So that's it is. So so other thing is, yeah, poly T tail you can use to isolate this mRNA. It's not necessary now. I'm not talking about that, but you can imagine the essential part. Promoter is essential. So this what is MCS? Multiple cloning site. Have we talked about that before? Right? What is multiple cloning site? It has large number of restriction sites. Large number of restriction sites. So we use EcoR5, right? So you use EcoR5 enzyme to cut this. EcoR5 enzyme to cut this. See? EcoR5 enzyme to cut it, right? So then it is open up. When it is open up, you get EcoR5 site here and EcoR5 site here as well. All right. So now you put your insert, your gene here, right? But you cut from EcoR5, right? So EcoR5 site here and another EcoR5 site here as well. So same matches are compatible. Edges are compatible. See? Edges are compatible. So you can imagine either this one to that one. That is one way of doing that. Or this one with that one. That is the second way of doing that. So we don't do that. They will happen automatically. So what is the enzyme? Ligase, right? Ligase enzyme will do that. So DNA ligase enzyme will, it is the only enzyme which can attach two DNA fragments together. Okay, remember the name ligase. So this one way or the second way is fine. But what if the vector self ligated, vector ligate vector, right? So without insert, that is not good. Understand? All right. So we come back to this. Remember this small bit. So we check what is the next step before we further discuss, right? All right. You go there and see what is next step in your answer sheet. But see, the answer sheet didn't mention about Southern blot at all, but they just say just pick up the insert fragment, right? But now you know how it is doing. So then you can actually add number of different sentences in between which carry marks, right? So Now, bacteria plasmid act as DNA vector, carries animal, that's the recombinant, So now you use it, now you ligate it, so it become recombinant DNA. But it has to express as well. If it is don't express for some reason, probably it is a mutated fragment, like fragment in PCR fragment, mutated fragments can come from there. So if it is recombinant, even it don't express, it is not the recombinant technology, right? So you have to make sure it express as well. But see, how do you identify, right? If the bacteria cell pick up, if the bacteria cell pick up the vector, right? So how do you identify whether that vector is, right? Whether that vector is good vector with insert, or vector is what is called self-ligated, free-form vector, right? So there is a separation of plasmid by centrifugation. Yeah, first of all, you have to identify the vector, right? It's a good vector or bad vector. This is a vector. Once it cut open by restriction enzyme. So multiple cloning site has equarphy here, equarphy there. So what if it is self-ligated, have no inserts, all right? No insert, self-ligate. But the second one, it has an insert. This is our insert. So this one go there, this one go there, and it will result that kind of fragment. So this is our insert. So there's no two DNA there. So this is not recombinant. This is a recombinant DNA, and this is not a recombinant DNA, because without the insert, it is just self-ligated, right? It is just ligate by itself. So this is a non-recombinant, not a recombinant. This is a recombinant. So how do you identify? Because they both have ampicillin resistant gene. See, ampicillin resistant marker was not in the insert. It was in the vector anyway, right? So to identify that, you have what is called blue-white colony selection, right? So blue-white colony selection. Now look at, I got that blue-white colony selection example. So meaning that, right? So you got, right? Once you put the vector into the bacteria, right? So we use the transformation, right? Vector putting into bacteria, what you call the transformation.